Yeah. Good morning, all of you. Please switch on your cameras, all of you. Akshita, Harsha, Naveen, all of you, keep your cameras on. Okay. First of all, I would like to congratulate you. Please keep your cameras on and make your mics off. Speakers, you make it off. And whenever you feel to interact with me, whenever you feel to discuss with me, please open your mic and discuss with me. It is an interaction kind of thing. It is a discussion based classes generally we take at microbiology. Okay. So see, the today actually in because we are having only online classes today. Actually, Wednesday it is a practical that you are having, but due to online system we are not taking the practicals. I'll be taking the theory class now. 2 hours, 10 to 12. I'm scheduled to take the class, and in these 2 hours, first hour is about general awareness, uh, teaching, learning goals, and aptitude. All those things I'll be discussing. And the second half, there is 11 to 12. I'll be discussing about in general microbiology. What is microbiology? What is the scope of microbiology? And uh, see this making you aware about the subject. Those things I'll be discussing in second half. So the first half is concerned, it is to just motivate you about the learning system. There is a teaching learning systems we'll discuss in the first half. So a majority of you are like here in Nizam College. Major like see, so far the cutoff is around 97%. All of you are creams and very good at your academics. I hope so, and you will keep it up your whatever your score is there in subsequent also. See here the intermediate and graduation plays very important role in life to settle down. This is very important thing. Intermediate, intermediate actually it is a basic pillar to decide whether to go into the professional like engineering, medicine, kind of thing. And afterwards the graduation plays important role in see majority of the jobs, even IAS officer, IPS officer, banking sectors, all the sector, majority of the police, majority of the sectors are being placed with graduation. So how you are doing at graduation plays important role in your life where you are settling down. Of course, see, you will be doing PG, and some people may be going PhD also, but the foundation laid at BSc level is a very important role to determine how you will be making your career or your future. So this is going to be very important role. 3 years of time. This 3 years of time is very important for you, and if you give it best, if you along with the subject, see nowadays you are learning online, and a lot of online materials are available. And if you along with the subject, just not the subject is enough. If you along with the subject, if you can learn some attitude and like some goals, there's a then it will be easy for you to settle down in your life. So that's very important thing. You should have an ambition, or you should have a goal for your life. So see, up to intermediate, majority of the student, 97 98% are possible with only Narayana and Chaitanyas. But IQ level, the problem of the Narayana Chaitanya students, see, this is what we observe regularly. The problem with corporate education, I don't want to name any person, but the problem with corporate system is making you by heart and putting it on paper, vomiting it on the paper. But finally, when you plan for reasoning, when you think about the subject, suppose amoeba is making the logopodius, pseudopodius, and it is useful for its food capturing and its see just how it forms, how it does. That is very well taught to you, and you have studied, and reasoning behind it. Actually, what is the metabolic process that is happening over there? And why it is happening? Neither the teacher is taught nor the student is asking, because you are asked to make you are asked to learn as it is. uh, just taking a subject for exams, that was happened. uh, See, in the schooling is concerned, will actually our Indian system schooling is excellent. We try to understand the concept, uh, because we don't see, we believe that teachers are the gods, they knows everything. And whatever the teacher that is delivering, school teacher, primary teacher, or school teacher, they are delivering the information, we used to believe them. And we used to grasp it. Even see, whatever we studied at school level, if a MBA person who has settled down as a tax analyst or somewhere, he will he, if he has studied biology at his intermediate level, he don't remember. But if he studied whatever he studied at school level, something, whatever the pollution or whatever the concepts are there, definitely he do remember, because he remember by heart that one. And now it is the time for you, you need to learn the subject for making your bread and butter. That is the most important thing. See, man, if something, if you are getting something, if you are going to politician or a bureaucrat, if you are expecting something from him, then what you will do? You will bend and you will salute to him, isn't it? Because you are having some expectations over there. So if you are having expectations with your subject, if you are having an expectation, suppose whatever the microbiology practical that you are going to do today, or the subject that you are going to learn today, if suppose you are thinking it will give you bread and butter, then you will be having a focus on it. You will be having a focus, then only there is a possibility of having a focus. Otherwise, just again the same thing, making by heart before the exams and vomiting on the paper, and after the exams is over, after getting a degree, zero subject. That should not happen. You should have the complete subject with you, not the marks subject. Generally, so far whatever you did is the marks subject. You tried like sperms fighting for ovulation. You try to score more and more. And see, whatever the people who comes to Nizam College, just they miss the medicine, and they are second to none. They are extremely good in securing the marks. I need not to teach that one, and I will not teach you that how to secure the marks. Here is concerned. We are here to teach you how to learn the subject and how to make that subject useful for your subject, for your life. That is most important thing. Learning this, that is a workable subject is important and marketable. And see, I am the placement officer also here. We do placements to students. We show the jobs to our students in various companies, MNCs, and different things. So see, you should make yourself marketable. You should be your degree should be sold somewhere. You should get bread and butter. Why you are studying? Is it fancy, or it is a hobby kind of thing? No, no. If you are telling that one, just to make you make me impress, I don't want to see. You are learning here. Your main goal of your life is first and most important thing, making your life successful. For making a life successful, you should have bread and butter and job, and the remaining things will come along with it. So a degree which does not gives you a job, a degree which does not gives you money, then it does not gives any sense, isn't it? If a degree with whatever 3 years of hard work, if you are not getting a job, if you after 3 years you will not be able to speak about your subject, or if you are not able to talk just 10 minutes on your microbiology subject, or the skills of subject, or any general, suppose you are learning English literature. If you are learning English literature and you have scored 99%, and for your kind information, see, many people who gets failed in this NEET, the main problem is making by heart and not taking the subject to your heart, not understanding the subject. That is the main problem. And I will tell you one more reason. IITs, majority of the IITs from our Telangana and Andhra, majority of the IITians are dropouts, because they are making by heart and writing this exam, and they are scoring good and getting the admission, but afterwards they are not able to understand the subject. They are just adopted for making by heart. They are not adopted to learn, to understand the consequences of the subject. What happens? See, after A, B, C, always everything will be in chain. After A, B, C, D, if the C is missing, he cannot go to the D. He will be dragged at B. He will be stagnant at B. This should not happen. Actually, I'll tell you. See, this session is to not only education about your life skills also. I want to give you some life skills also. You must have seen several. It is unfortunate, but I'm starting in the day one. Many suicide incidences you must have seen. Student is committed suicide, or that IIT engineer or software engineer or somewhere has committed suicide. Recently, it is becoming very high. What could be the problem? See, the problem is, see, nothing is end at our life. If something happens, if you lost money, or if you lost your friend, or if you lost your parents, or if you lost somebody, that is not the end to your life on that particular day. See, what happens if you learn the studies just by making by heart? If you are this what happens more in case of the engineers and mathematicians. Mathematicians, they are supposed to make by heart of equations, formulas, and other things, and that is not happens with biology students. And see, when they are doing the interpretation, if something is when they are making a by heart, if something is missing in between, I told you, for doing a reaction, A to B should go B to C, C to D. So if a fellow, the person who makes by heart, if he missed C, then he is not getting the conclusion of D. He is not having the ability to interpret. Reasoning is not there in that case. If you whatever the way that you train your brain, it will take in that way. So majority of the computer engineers, or there's IT people, or mathematicians, they are more trained in making by heart. So it is very unfortunate. They will not do, if suppose some emergency has occurred, somebody like somebody is troubling in office, or love failure, or they didn't get the increment, or whatever. See, life is full of problems. So if you have a problem, see, you are all biologist. Nerve stimulation takes place continuously in our brain. Nerve stimulation, it is a continuous process. Signal will be transmitted continuously. Whereas if you are if you are a there's a by-hearting kind of candidate, then the signal will be passed at one particular area. The signal, see, he has stopped at C, he has stopped at B. He's not getting the, he could not able to recollect the C. The ultimate goal is D there in that case. See, there's one girl, she didn't get increment, or she got a love failure. So her ultimate goal is getting married or getting a boss chair of that office, but that was not happened. So she was not trained to do the reasoning. She was trained to do the only arithmetic. What you do in case of your intermediate, just making by heart, by heart, by heart. So what happens here? She could not able to do reasoning. See, there are why I could not able to break through in this one, and what happened, and that she will not do. Just she will do that. She will think that I could not able to go to the D because I am an inefficient. I don't know the C. So the signal stops over there. Nerve signal or nerve impulse transmission gets stopped over there. Whenever the nerve signal transmission stops over there, then the entire brain starts working to resolve that issue. Entire nerve system, your brain completely. So it requires a lot of energy, ATP. A lot of ATP is required, and see that leads to the stress. That leads to the stress, and these stress, like most of the time, those candidates will not able to tolerate, and they commit suicide. There is a simple logic. See, have you seen any news any apart from the engineering stream, apart from that IT stream, other students committing suicide? There are maybe, but very few, because you should be trained yourself to reasoning, not making by heart. And if you are reasoning, it will be stored in your permanent memory, and you need not to by heart, and you need not to struggle for passing the exams. I'll tell you one more reason. See, in MBGC, microbiology, genetics, chemistry, so far the cutoff marks is around 97%. Maybe some reservation categories, something is maybe there. So this 97%, and if you see the degree examination pattern, you will be having 8 short answers, out of which 4 you are supposed to write. In each unit, you will be given 2 short answers, 2 long answers, out of which 1 short answers and 1 long answer is supposed to be written. Means you are having 50% choice, isn't it? 50%. And see, the syllabus is concerned, chemistry, almost 50% of your intermediate syllabus will be there. And see, you have secured almost 98% in your chemistry, 98 to 100%. And it should not be a problem for you to get 40%. Hope so. I asked you, all of you, keep your cameras on. So when you secured 97%, 98% at your intermediate level, why you are facing the problem to get 40%? Majority of the students are facing the problem to just pass the degree. Why? What could be the problem? Because you are still in the same socket. You are trying just again. See, here now you are in the semester system. Semester system will just move like 3 months, 2 months. There will be a classes. Then immediately you will be having a practicals. Then there will be a semester theory examinations. Then again you will be having the theory, 2 months theory classes. Then 1 month practical exams. Then again theory exams. This will be the cycle. It will be a fast cycle. And you feel that like almost I feel junior college is like a jail. You are in your principal or your college, the people are making you to sit in whatever the jail is concerned, and you are not free, you are not allowed to see outside and outside environment. But here, when you come to the degree college, you people feel that, oh, I have grown, I'm grown, and I can see enjoy the society, and I can enjoy the environment. You feel like that one. There is no doubt in that one. You are grown up, you are doing graduation, you are grown up, but responsibilities do have come on you. A lot of responsibilities do have come. See, you actually, a best person sees the environment and chooses the best. You see what is there around you, what is there. Nobody can prevent you from the environment. And a lot of sources, almost everybody are using Android phones nowadays, isn't it? You are using the Android phones. Android phone is good as well as bad. So make use of a good side for you. Make use of good side instead of sitting for just playing on games and just watching unnecessary things. Make use of search your subject. What are the new things that are coming in your subject, and what are the new areas that can be still improved. Just take it as a challenge and you work in that line. Then it is a possible of making there's a Android phone. There's a, see, because of online education is going on, each and every person should have Android phone now. But make use it in a proper way, not in misusing it. You must have seen there's one PUBG game. Because of the PUBG game, several people have lost their lives. What could be the reason? Can any gentleman tell me why XYZ X would have lost his life in PUBG game? Nikita, Deepak, somebody.
Maybe lack of responsibility, sir.
Okay.
They don't know what to do in their life. They're just
Yeah.
enjoying, and someone say you need to enjoy and start your life. They feel bad and they're going to commit suicide.
Is it so? Life is that so easy? No, it is not. See, I told you the first example, suicides in IT people. They don't think anything in the future. If I could not able to achieve, I am the failure. This is the main reason. If somebody makes you to feel yourself bad, there a person will not think anything. Always see this entire story I am telling you to mentor you. See, some of you must be having a bad impression that I lost medicine, I lost MBBS. MBBS is not ultimate. You just go somewhere in corporate nursing homes or corporate hospitals. There are some duty doctors, junior doctors with MBBS. They are doing vital checkups like BP and temperatures, all these vital checks. They are doing, and they are being paid 5 to 10,000. Do you think that MBBS is successful?
No,
It's
sir.
not. It's not. Uh, Anyone, see, even if you are taking the BA history, HPP or something, if you are doing it, if you are giving your life complete potency into that one, and that is also more useful. And see, it is not, and microbiology is a subject where you can do wonders. I'll explain you in second half. First, let me explain you about teaching and the importance of teaching and learning processes. So, see, my conclusion of the entire story is we should have a reasoning based teaching as well as learning. We should not make by heart. That is one. Second one, you should have a goal in your life. Why you are learning, actually, why you have come at 10 o'clock sharp, you have like you joined through the link, and you are facing me. Why? Because you are having a goal, you want to achieve something, but make it clear in the beginning itself what you want to be. If you want to, so see, life should have a long goal and short term goals. Life without goal, just see, just if you want to pass, getting past BSc is a short term goal, not a long term goal. And whoever joins the BSc, definitely he will pass. There is no issue at all. And see, I'm here in teaching field since last 20 years. I have never seen any students, especially Indians, failing in microbiology. Nobody will fail in microbiology. And MBGC students will not fail, especially Indians. See, we are having some foreigners also. Foreigners, generally, they don't have the subject. These subjects, chemistry also, they don't have, or some applied subjects are there. Dairy chemistry or agrochemistry, such kind of subjects they will be having. So they will be facing some problem. Even the language communication problem is also there with them. So they will face some problem. But here, the passing is not a problem for our people at least. So you should have the goal for making your life successful in the sense getting a good position. Suppose I want to get a secured life in IAS or IFS, IPS, or per se I want to become a great scientist, or I want to become you choose your the goal. See, we are here to mentor you. Actually, the best teacher should be a best friend. He should not, he or she should not pose his interest or his choices on you. He or she should facilitate your goal. Suppose Gautam wants to become an IAS officer. So what, as a teacher, I should help him to achieve his goals in an easy manner. Whatever the things, see, if I can do something, I should do. Otherwise, I should send to a person where he will be getting motivation, or where he will be getting knowledge about the subject. That we will do at microbiology. Always we will be mentors than that of teachers. We will be facilitators to you. We will be able to tell you which is a good and bad. What is a good and what is a bad for you. And I hope that you will choose the best. First of all, and we definitely will help you the scopes in the afternoon, like in the second session, from there's 11 onwards. I will tell you what are the scopes of microbiology, so that you may be able to make your mind clear. So because at intermediate level you must have studied microbiology, but very little. Okay, so that I will make you clear about it. So first of all, you should make your mind clear what you want. And see, never underestimate any other person. Always somebody will be having a edge in particular subject, or particular art, or particular there's a quality, isn't it? Always, so you interact with other persons, whoever comes in your life, and try to get the subject from them, and whatever that is possible, you try to gain from them. And see, this is possible when you are making your mind open. Some students, what happens, they makes themselves close within a circle. They think that that is the world. See, now earlier subject wise research used to be done, like only biology people used to do the biology, chemistry people used to do chemistry. Now it is a intradisciplinary. We are working physics work also, chemistry work also, even some parts of economics work is also being carried out by biology people. So it is not now it is an interdisciplinary area. So you interact with as many people as possible, and you should have a goal in your life. You try to always see, that is my goal. So how this is connected, how this topic, or how this fellow, or how this whatever the environmental observation that it is there with you, that you are observing, you try to correlate with it. Then the learning will become live and more interesting. The learning become more interested when you add your experiences or observations, that you are interacting with the subjects or persons or environment, with your goals or what you are having. Then only it is possible to make it lively. Then only, see, when suppose I'm teaching about ultrastructure of bacterial cell in a class, I am teaching myself or some of my other colleagues, we are teaching ultrastructure of bacterial cell. So we'll be explaining like one by one layers of bacterial cell, we'll be explaining. So if you are live, if your ambition is to become a doctor, suppose a scientist, if you are so if you want to become a structure, when the teacher is explaining one particular structure or one particular layer, you should interpret. So if I damage this structure, the bacteria may not be growing. Then I will be giving a treatment, a new treatment or new strategy to the disease of this particular bacteria. Then you will become a great scientist. Then it will become a live subject, or it is called as working subject or marketable subject. When you are involved it, whenever you are involved, see, teacher says something, you are interpreting out of it beyond the teacher's presentation. Got it? Your teacher is explaining see, sometimes teacher also going beyond the content, but see, because we need to cover the syllabus, several issues, and see, you are having huge syllabus, 4 units are there. I'll share you today the syllabus also with you. So as we need to cover the syllabus, always out of box thinking may not be possible. So while learning, if you are live in the class, then when teacher is explaining one particular concept, you are supposed to think out of box. So if I do this one, if this is happened to be like this, hope you are getting my point, Deepak. Okay, so
Yes, sir.
then you will remember the subject for your life period. This is possible to remember. It is not making by heart, it is reasoning. This is called as reasoning. When you are interpreting, when you are thinking, this is happens when I do like this is called as interpretation. This, there the life starts successing. Your life will become successful when you are doing the interpretation. And I will assure you, Deepak, make your phone off, there's a mic off. Okay, I will assure you, if you start reasoning, it is called as, see, reasoning or interpretation. Reasoning is thinking around the subject. Thinking around the subject, I will assure you, none of you will commit the suicide if you do the reasoning. You will never stuck up at a problem. This is research based finding, I am telling you. If you are reasoning a subject, you will remember, you will be live in the class. It is a glowing face you will be having in the class. It will not be dull. So your brain will be continuously will be taking the message and it will be interpreting inside. Reasoning will be see, brain will be having two sides in our brain will be a right side and left side, and right side is responsible for interpretation, left side is for arithmetic reasoning, arithmetic remembering. So in case of scientist, right side brain, especially biology people, right side brain is very much developed. And left side left side brain is more developed in the people like computer science, where mathematics or history people, where they need to remember, they need to make by heart of all those things. And the people who where the arithmetic brain is more developed, they are prone for this committing of suicides. Whereas right side, if you are good at reasoning, you are very good. See, you are the best person to give the solution of common societal problems, or you are whatever the environmental problems. You will find out if your reasoning mind is very good. Just so, if you train your brain in interpretation, that will make you successful man in your life. Not only that one, in your subject, you will be live at your subject, maybe idiot at your intermediate level. See, getting 90% may not be, see, to say that the person is intelligent or super intelligent, but there just you are making by heart and putting it on the paper, and they are giving some questions also. I remember, just they are giving the questions and you are supposed to remember those questions and write, but that will not help you to become successful man in your life, successful person in your life. So first and most important thing is be analytical, be reasoning, be interpretive. Whatever the class goes on discussion, you do the reasoning of it. Just see, you there are some situations. Ex-scientist born in 1647. We cannot do reasoning in this one. Everything is not reasoning. Somebody is born, somebody got Nobel Prize in that year. That is just not reasoning is objective base, subject base. Okay, for your technical subject, even English, if you are learning, suppose a teacher is explaining the grammar, try to, whatever the grammar she is explaining or he is explaining, make use in your life by practicing or using it for your conversations. Then the subject will become live subject will give you a lot of benefit. And second, one more thing, in this learning process you should have a best friend. Actually, everybody will be having friends in their life. We'll be having schoolmates and college, junior college friends, degree college friends, PG friends. You'll be having several friends, but generally, because this is some extent matured age, isn't it? So you'll be having more friends over here. And here, see, that is the friend is the only gift that is given by God. See, father, mother, rest of the things, everything is given by God. But friend should be, you should have a best friend. Friend in the sense, the person where we can believe. Tell me just who is the best friend. I want to be a best friend with you. Tell me what should be the features of best friend.
Being transparent, sir.
Being transparent, and
Should help his
Caring like
friend.
a family member.
friend in deed, a friend in need is friend in deed. Is something like that one. Okay, whatever may be. See, this is not the session to go in. Some other time we'll see about it. Actually, everybody will be having, there's a flaws and there's something good and bad in each and every person. So actually, the friend should whatever the exact, without making it high or making it low, as it is. If somebody is showing, that is a great friend. Something Deepak said, transparent. See, in that context, actually, mirror is a best friend. Mirror is a best friend. Whatever, however you are, when you stand in front of a mirror, it will show you what you are. It will not aggregate, it will not disaggregate, it will not enhance, it will not reduce. Whatever you are there, that will be shown. And one more beauty of friendship is, that is the quality of mirror. It will not say what you are to somebody else. Lot of people stand in front of mirror, isn't it? Does it say like, how is your mother, how is your sister? Is it it will not reveal you. A best friend, suppose if X is my best friend, X will tell to me only whatever the things that I would like to share, that it will share with me only, not with somebody else. That is, you become a best friend first. If you are expecting somebody to be a good friend, first you prove this rule. You should not, there's a enhance or whatever, you should not, whatever the qualities are there, just you show those qualities. Okay. And see, actually, you need not to help. Actually, indeed is a need, and is indeed when you are supporting in time, When you are showing what he requires, then he will do him by himself. You should make your friend self-reliant, not depending on you. Even me also, as a friend of you, my job is making you self-reliant. What do you mean by self-reliant? What do you mean by self-reliant? Amarnath? Amarnath? Sleeping, huh?
Yes, sir.
What do you mean by self-reliant? Reliance. Nowadays, lot of many
Self-defense.
self
Self-defense, sir. మన
self
మీద మనమే
dependency. Yeah,
మనం
self dependency. Atma stairyam, Atma nirbharam, Atma nirbhar. It is in Hindi. Self reliant. So we should, if you are becoming self reliant, you are the most successful man in your life. You should become self reliant. You should not depend on somebody. You should not depend on Dr. Pasha for your subject. I will make you as such that you should become self reliant, and as a teacher it's my job to train you in that context. Okay, so for which you should be a reasoning mind, you should be open mind, you should have a goal in your life, and you please discuss with me what you want to be. Anyhow, maybe it may be taking 15-20 days for offline classes. By that time, anyhow, you will be learning, you will be understanding what is microbiology, what is genetics, what is chemistry, and even other subjects also. So you make your mind, and we are here to help you to achieve your goals. It is not getting a generally what I say is, if you miss the doctorate, the stethoscope doctorate, you should target, if you are very good at microbiology, if you are very keen at microbiology, if you want to settle in microbiology, you should target the doctorate of microscope. That's a PhD in microbiology. We call, if you miss the stethoscope doctor, then you should become a doctor of microscope. Actually, this is millions of times better than that of stethoscope doctor. That you'll feel subsequently. So and the relation between the student and teacher. Actually, instead of teacher, I'll be happy to be a friend, a mentor, a facilitator. A friend. My job is to identify the what are your pluses and minuses, and express only to you what you are, so that if something is minus, you'll remove it. If something is plus, you will improve it. That is, I want to be a friend of you. Second, facilitator. See, the content is available. Content is available. Huge content. If you many microbiology. In fact, I have written 25 microbiology books, and see, all these books are available in there is the internet. All these books are available. In fact, you can directly go to the internet and you can collect the information, and without Dr. Pasha's requirement, you can study. There is no subject is not the intellectual property right of Dr. Pasha or XYZ, isn't it? It is available. It is the school days. We feel that teacher is a god. He's having a lot of information. He's sharing. But now I'm not telling that I'm having the information. It is already established one, and just we are sharing with you, isn't it? A facilitator facilitates its a proper understanding. Just whatever you are trying to understand through online or through e-content or through books, whatever you are trying to understand, just will facilitate, will try to make it easy. I'm not giving something new to you which is already existing. I will try to make you to understand in an easy way. Okay, especially the applied subjects like microbiology and genetics, they requires a lot of skills. They require, see, suppose you are standing on a swimming pool, and you have studied one book, one complete book, how to swim. You have studied one complete book. Are you able to swim? What, Nikita Reddy?
No, sir.
Why?
Because we should have try once.
Once. So once, suppose if you are trying to dip inside, go inside, you will be sinked.
Yes, we
What I
should
want to
have
say,
help.
yeah, no, no. It is what I want to say is skills are important, ma. Here,
Yes, sir.
skills are important, not the theoretical knowledge. So applied subjects for to make use of subject in your life, you require the skills.
Yes,
So
sir.
a teacher is not only the knowledge, teacher is actually facilitator, provides the skills. Facilitator provides good skills to you, how this particular thing. And one more thing, a teacher is a, see, a lot of things happens in microbiology or in general life also, a lot of things do happen. So he's adding his life experience to your subject. A facilitator makes you, tells you how this can be done in a easy way. Suppose, see, Western labs, most of the Western labs, like American labs, they are automated. You must have seen pipette. Have you seen pipette?
Yes, sir.
Normal pipette, which you must have used with your mouth or with a bulb. You must have aspirated some liquid. Huh.
Yes, sir.
But in Western countries, they don't use pipettes. They will be using sophisticated micropipettes. Sophisticated micropipettes. So the fixed quantity can be drawn from it. So suppose if there is an instrumental problem, if there is an issue with the pipette, the micropipette, then they are supposed to stop there itself. The work has to be stopped there. But see, we are the Indians. What we'll do? We'll definitely will find out a way. This called as jugaad or alternative, isn't it? We'll find out a alternative so that work will not be stopped, isn't it? How this comes, alternative or jugaad, how it does it comes? That is with experience. That comes with experience. Just if you are going, just routinely, if you are going with the subject, just paper subject, if you are going, that is not possible. So we should be prepared. If the routine things, if the main thing does not happen, we should be prepared with the alternative things. Actually, many of the innovations or discoveries happens because of this reasoning knowledge of jugaad or alternatives. This is very much required. And for applied sciences like microbiology, especially for microbiology, you require a lot of skills. And a lot of skills in microbiology. In fact, see, up to degree, like I have not studied microbiology. MSc and PhD I studied microbiology. So microorganisms, the bacteria I have seen in MSc first time. I have not seen, never seen the bacteria before that one. And microscope also, there is a cross section for botany experiments we used to observe, and some spots we used to observe in the section cuttings we used to observe under the microscope. We have not observed majority of the things. And you are lucky enough, you are learning the microbiology at your degree level. So what happened when we were doing the higher, the MSc, majority of the students who joined along with me, my classmates, they were from BSc microbiology. So they are having a lot, like already 3 years they have learned the subject, microbiology subject. They are very well versed about different techniques of microbiology, and we are not aware about these things. And we used to feel shy, how I should ask. See, nobody is asking, no students is asking to teacher how to do this and that, but we are not aware. So what I used to do is I used to just some attenders used to be there, even you will be having. So the skills, we should not feel shy to learn the skills from the whoever is there in the lab. And many of the important microbiological skills that I have learned from the laboratory attender who used to be with us during our masters in microbiology. So that comes, see, not only the bookish knowledge, like swimming, the learning in the swimming, the skills you will be learning, whoever are there in that swimming process, maybe a child, uh, uh, the small child in Western countries, like beaches, everywhere, every people like from 3 years, 2 to 3 years, they will start swimming. They will go to the beaches and they will start swimming. So we don't have much swimming practice. So one of my friend has like it was happened to him to go to the beach. So a small girl of probably 5 6 years made him to learn how to swim. It does not require a professor or a trainer in swimming may be required to you to train in that particular subject. Even in microbiology also, to train you how to prepare the petri plates, how to prepare the plugs, it may not be the professor Dr. Pasha, head department of microbiology required. Even the attendants who are working in your lab, they are maybe see here what I want to emphasize is the experience matters. What they learned through their life process is very important thing. And this also you are learning. This is also you are learning. Okay. So I want to be see my team. We are having 3 members here, faculty, teaching faculty. I'm head department of microbiology, and there are 2 other members. There's Dr. Anju Rajan and another Dr. Hanumalal. All 3 are PhDs and good qualified. And when you compare with other departments where all PhD qualified, and we do maintain strict classes, no classes will be missed, and we expect similar response from you. There is no leniency, we will not be taking any leniency. And see, you treat us as a friend, no formalities. Sir, may I come in? Not required. We know that you are there's a goal minded, you are having a goal to achieve. Suppose if you are going for civil coaching somewhere else, we do understand, this Gautam has to attend that class, so he's coming late. We don't mind anytime. In fact, we'll give some attendance if genuine person if he's doing. And if somebody is having really financial requirements, if some, definitely I will help them to do some part-time job. I will arrange some part-time job if really requires. See, I told you I'm your friend. So definitely we are here to understand your requirements, and try to see, it may not be the 100%, but whatever the way that is possible, we are here to solve your problems, whatever the way that is possible. So you should take it in a positive way. And see, even the seniors, whatever the your second years, third year students, what we have, you need not to worry with them. Many students and parents get scared of ragging. See, first of all thing, I'm proud to say that microbiology department is subject or career oriented subject, and students are always focused, and they will not be unnecessary involved in such kind of ragging issues. But see, I cannot expect all the microbiology students only to roam around the college, isn't it? There will be some other people also. Even in microbiology, there must be 1 or 2 exceptions. Normal distribution you must have studied. Distribution of students, see, 10% will be extremely well, 80% will be normal students, and 10% will be poor students. So, see, this 90%, 90 to 95% students of microbiology will be very much focused. They will be always out of box thinking. When you see the microbiology lab, you must have seen the 3 Idiot movie, and the lab established by Amir Khan, and which is innovative, doing something new. You will enjoy the same atmosphere when you come here, but at larger extent. Our students do get patents at BSc level. I'm having 17 patents, 25 books, and these patents, see, the students will be doing a lot of innovative works, and their works, students' works, even they gets commercialized. There are many. Okay, so when you are focused for something, when you are thinking of innovation, last year seven students have participated in Telangana Innovation Yatra. Their innovation projects have been selected, and last year we have filed 4 patents. That is possible when you are focused, but see, maybe 1 or 2 persons may be interested to find out you. Obviously, there is a curiosity, no? He may be interested to find out who are you actually, where you are coming from, and what is your background, all those things. He may be interested, but it does not mean that he is doing ragging, isn't it? And see, always make a note of my number. Always, just anywhere, not only the microbiology, anywhere, if he's an art student or commerce students or economic student or whatever the student, just tell him that I am the student of Dr. Pasha. I am having his number. I hope that he will not trouble you. Actually, our students are very focused. They will not involve in any ragging kind of things, but maybe 1 or 2 candidate, just he may be interested to find out your CV or name, from where you are coming from, and what some sort of information that he may be seeking. Because why his seniors were asked those informations to him. It is coming like a hereditary kind of thing. So for which you should not take it as a serious, and at the same time you should not respond in an aggressive mode. Isn't it? If you're see, it will become a there's a status. The problem will become why he has responded in that way. You always, when a person is polite, when you are focused on a particular goal, you will not be unnecessarily arguing with other persons. If somebody asks, what is your name? I am Gautam Kumar or Amarnath. Whatever your name is there, just you explain him. Nothing to worry. If he is making you to sit, stand or something, just all of you should make a note of my number in your phone and just give a call to me. Okay, I'm the senior faculty and I'm research committee member and placement officer, head department, even genetic. I used to take care recently, and now we have given to them. So there will be no problem at all. All of you, just I will just make a note if you are having a book or some 944 103 1626. Anyhow, it is there in there is our group also, WhatsApp group also. Just make a note and see. I'm telling you, there is no ragging issue in microbiology. I'm talking about in our area. This is some of you must have seen the department. It is just adjacent to our gate number 2, just at security, just adjacent to the security. This is bit isolated. It is separate from other streams. Genetics and microbiology, these two are adjacent, side by side. There is no problem, no issues, no ragging. And if somebody parents, especially girl parents, are scared of ragging issues, see, we are here after, anyhow, you are the parents. We are here to take care about your children more than that of you. You need not to worry. We will take care, and generally there is no issue at all of ragging. And when you handle the situation in a proper way, when you are not responding, see, if they are asking the name, just give them the name. Just whatever the details, if they are asking you, just tell. But don't panic over there. If you start panic, if you are fearing over there, then they will start more. They feel that they have won. That should not happen. Even if you go to the second year, you will you may be also interested to find out the names of your juniors, isn't it? In fact, your seniors asked me to add. See, we are having a Nizam College microbiology group where all first, second, third, third year students are there. Even pass out students are there. I have not added you people into that group because it takes some. You people may be scared. So let's wait. You will get that strength in due course of time. So don't worry about the ragging. When you are strong enough, Nothing is going to happen. Don't worry, and no ragging will be taking place. This is a direct message that I'm going to give to the students and their parents also, especially girl students. And so be focused. You make your goal. There should be a long-term goal, and you should have a short-term goals. And short-term goals are adjustable. They should be changeable. They should be. You should not fix that. No, no, no. I will go in this way only. That should not happen. Okay. And we are here. I am here, and my staff. We are here open from 8. I will be coming to department because of now, because of lockdown, and this students are not coming. We are coming around 9 o'clock. But normal, my lab working mode is 8 to 8. So you can enjoy. And microbiology is a research-based subject, skill-based subject. You can do wonders. As I promise you, after mentoring, I will start now the microbiology. So the microbiology is a wonderful subject that you should feel anyhow. See, just by taking a pinch of soil, you can collect millions of microorganisms out of it. And the microorganisms are there. You name any function, they will say, I am there. All different activities are being carried out by the microorganisms. Microorganisms, what is there in your mind? Microorganisms, in generally, forget about your student. In society, what the society thinks about microorganisms? One by one, Deepak.
Few people think they are harmless,
Okay, Gautam.
and some people think they does not exist.
One by one. I want everyone to express their view.
So they are helpful. They are helpful to in many ways, like in making curd or in making antibiotics.
Okay. Muzammil. Okay, Shiva Prasad.
Yes.
Yeah, what do you think? What is your opinion about microorganisms?
Microorganisms are present everywhere. They are small. They cannot be seen by
No, I'm not asking. I'm not asking the subject. Okay, make your mic off. See, normal people, you have studied something about microorganisms at your intermediate level, but normal people, if a layman, to your parents, if you ask about microorganisms, what they say, immediately the first opinion that comes, microorganisms are harmful. Immediately, the common man thinks that microorganisms are harmful, germs, isn't it? Majority, see, you have studied their beneficial roles to your intermediate level, isn't it? But normal layman, normal person, generally they think that microorganisms are harmful. Bacteria, you say that bacteria, oh, dangerous, they causes the diseases, they are harmful, isn't it? This is the feeling of common man, isn't it? But see, that is not absolutely wrong, that is not absolutely right. There are few microorganisms. See, now because of this corona, novel coronavirus 2, NCV, lot of people are thinking about microbiology, even virology, about viruses. They are thinking, earlier virus means the only some program which damages the software. See, we know virus, man. As a biologist, we are aware. But normal person, I'm talking you. Many of them, they are not aware what is virus. Because of this disease, it is only one virus. It has created such a huge havoc, and that virus has made us to come in contact with online classes, isn't it? And lot of GDP has gone down, and many countries, their economical situation has become very bad, isn't it? That is the impact of microorganisms. Generally, the people think that microorganisms are harmful, they are dangerous, but very few microorganisms are harmful. very few microorganisms, and majority of the microorganisms are useful for us. Majority, very few are, but see, even though there are very few which are harmful, because due to the severity of the problem, we used to think that everybody is bad. Suppose in one class, one fellow is making a nuisance, only one fellow. Huh? Because of one fellow, and he is making a huge nonsense over there. Whoever the teacher goes to that class used to name that class is nonsense class, isn't it? Microorganisms, there are very few, we can list them on fingers, which are pathogens. Very few microorganisms which are pathogenic. But majority of them, we cannot survive on this earth. Without microorganisms, we cannot survive. That is fact. You name any activity, microbe says that we are here. Cleaning of environment, digestion of cellulosic material in your intestine, and there's a prevention of toxicity by chemicals in your gut. All these metabolic activities are being done by, see, the lactose. You are from childhood, we are drinking the milk. Everybody drinks the milk. So this drink is having lactose, and lactose, none of the human gene is having to metabolize this lactose. We cannot metabolize this lactose, and we cannot digest this lactose. And we are thankful to E. coli, Escherichia coli, which is capable of digesting this lactose. And what happens if it is not digested? If we are not digesting lactose, what happens to us? Suppose E. coli is not there. E. coli is not there, and it is not digesting lactose. What happens to us? This is the analytical study, reasoning study. What happens?
Children does not get energy from mother.
Okay.
It's get accumulated and it can be toxic.
Okay. Next. Okay. See, it gets accumulated as it is not digested by human genes. Human cannot absorb and digest the lactose, and it gets accumulated. As it gets accumulated, what happened? Osmotic pressure gets increased. See, like you are having a eggshell, which is egg membrane, and the inside you are adding the osmosis experiment. You have learned, no? Osmosis experiment.
Yes, sir.
In your botany, I believe. So you are taking an egg. Your stomach is like an egg, which is made with a membrane. Membrane is nothing but like your egg membrane. Here it is a cell wall, and when the lactose is increased inside, when the lactose is increased, what happens here? Osmotic pressure increases. In order to balance the osmotic potential, the water from outside enters into inside the shell, that is stomach. Then water comes from outside and accumulates here. As the inside water content increases, what it should do? It should come out. That pressure should be released. That comes out. See, from when the stomach is full of water, it comes. Sphincter is there is a wall in esophagus. It gets opened, the sphincter, and it comes out in the form of vomitings. This is what happens in children, small children's who could not able to. Generally, after taking the antibiotics, E. coli gets killed in their stomach, so that lactose does not gets digested, and that lactose gets accumulated in the stomach, and it traps the water, and that excess water in the stomach gets coming out. Have you seen the kids doing vomitings? Small kids. That is because of indigestibility. That's the absence of bacteria in the stomach, and this problem is called as lactose intolerance. This is called as lactose intolerance. Even some adult people also, some people will not digest this lactose because of lack of E. coli in their gut, and this is very common in Western countries. And because of our environment, a lot of E. coli is there in our gut, and we are happy that lactose gets digested. And a similar problem you must have seen the pregnant women doing vomitings. Have you seen?
Yes,
Good.
sir.
Why they are doing? It is not the case of lactose intolerance. What could be the problem? Naveen Beshweni. Somebody, Akshita, tell me. Tell me why during the pregnancy period, some women do the vomitings. Why? Your voice is not audible.
Is it audible now?
Yeah, now it is clear. Tell me, what could be the reason?
It might be because of the indigestion or.
Yeah, see, generally they will be taking a lot of iron and calcium, ferrous, iron and sulfate, and these they should be actually iron sulfate should be digested into broken down into iron plus sulfate forms, and once iron is there is a ferrous or ferric molecules are there, they only will be absorbed. Iron sulfate will not be taken up. So some tablets, see, iron ascorbate, you just see some small exercise. If somebody is there pregnant at your home, just see the tablet. Some tablets, government given tablets are iron sulfate, ferrous sulfate. Whereas private, whenever you are getting iron ascorbate or iron citrate, ferrous citrate or ferrous ascorbate. See, dissociation means separation of iron plus sulfate is very less. Whereas dissociation of iron plus ascorbate, iron plus citrate is very high. So if you need to absorb by your tissue, it should be first dissociated. Is it true? That's true. If it is not getting dissociated, it is accumulated in your stomach. If it is accumulated in the stomach, osmotic pressure will increase. If the osmotic pressure increases, what happened? The membrane which works as a semi-permeable membrane, it gets a lot of water and puts in your stomach, and that lot of water comes out in the form of vomitings. Okay, ma, I am expecting you to learn the subject in this way. Okay, now let's come back to the microbiology. So microbiology, see, in last last 20 to 25 years, almost all Nobel Prize, it may not be to the microbiologist, but to the microbiology work it has gone. Either it is a chemistry, or it is a physiology, or any work, it has gone to the work of microbiology. There are many microbiologists who are awarded with Nobel Prize, but the techniques of microbiology are very important. Actually, they gives a pathway to understand the genes. They gives the pathway to understand the metabolism. That gives a pathway to understand any kind of higher things. Microorganisms, say, are the techniques based on the microorganisms are very important. They plays important role. Microorganisms, why microorganisms only? Why not elephants? I will study on elephants. No, I will study on bamboos, and I will study on bamboo trees, or I will study elephants. Why I should study only the microorganisms? Why it is advantageous to work with microorganisms?
Because they can live in our body system.
Okay. No, for studying we cannot study on the body, but see, they are very tiny, so that millions of bacteria you can hold in a test tube. Small up and off. There's a small up and off. You can hold millions of bacteria at a given time. Can you hold millions of elephants in a time? It is not possible.
No,
okay, so you can catch hold. You can study on millions of bacteria, as they are very small. You can study the number of bacteria, many number of bacteria at a given time. Second, that does not demand. See, suppose if it is an elephant, you need to feed a specific feed for it, isn't it? If you are growing a dog for your home, what you require some specific, it's a specific demands you need to fulfill. Some pedigree, or some meat, or some non-veg you are supposed to provide for it. But the bacteria never demand for its specific feed. It never asks for KFC for you. Whatever the damn thing that you are giving, it will grow on it. That is the beauty of microorganisms. It will grow fast. Elephant, it gets maturity up after 30 years. Then within 3 years cycle, it will give birth of one baby elephant. So in an entire life period, it may giving one pair of elephant may become hardly 6 or 7 new offsprings may be produced. But one bacteria within 20 minutes, Escherichia coli, within 20 minutes, it will become 2. Very fast and rapidly growing. Within 20 minutes, just within 20 minutes. See, it is very tiny, very small. So it can be easily handled in a small test tubes. It can be easily handled. And second thing is, it is easy to handle. See, if it is an elephant, sometimes it may be friend, sometimes it may be doing the nuisance also. Sometimes it will disturb to your its there's whoever maintaining it also. But see, the bacteria will not do such kind of things, and they will divide very fast. See, they are small, but the enzymes are, their metabolic enzymes are almost similar to man. Whatever the hexokinase is there. Hexokinase is the first enzyme, actually very important enzyme, glucose to glucose monophosphate production. In energy production, first, actually we consider as a first step of metabolism, glucose to glucose monophosphate. And this enzyme is same in bacteria. You take any bacteria, that is the same. So the enzymes and genes are almost same in bacteria and human beings. So if you want to study the hexokinase, you need not to get the gene and enzyme from man, and you can directly get from a bacteria, or you can grow many bacteria and you can get that enzyme and gene and you can study. Is it easy or not? It is very easy. Okay. And see, only few genes are there and all the genes are working. In microorganisms, there are only few genes are there. Like if you take the example of Escherichia coli, in human beings, how many genes are there? In human beings, how many genes are there? Nikita Reddy?
12 pairs, sir.
12 pairs. What is the 12 pairs, man?
12.
Amma? You must be talking about chromosomes. Chromosomes are how many? 23 pairs, not 12. Okay. See, there are many genes, but characterized genes are 30 to 35,000 genes are there. Whereas in bacteria, you don't have those many genes. And here, the non-coding region is there in eukaryotic organisms or human beings also, junk DNA we are having. Whereas bacteria don't have that one, isn't it? So it is very easy to study the genes. Complete, see, you must be not much aware about genetics so far. So we'll discuss later. Beetle and Tatum, Neurospora crassa, they worked. One gene, one enzyme concept. So that was Neurospora crassa is a microorganism. So all the concept of microorganisms like structural genes, regulatory genes, lac-operon, everything was worked out with the microorganisms. Why first in microorganisms? Because it is very easy to handle them. It is very easy to understand them. The microorganisms, it is very easy to understand them. Okay, and there are very few genes in them. So, like, see, if you take man case, suppose the skin color, many genes are involved in this case. Whereas if you take the pigment of a bacteria, if a color of bacteria, colony of bacteria, only one gene is responsible for it. So their understanding of one particular function is very easy. In bacteria, so generally, whenever you are being trained, you will be trained with these easy practices. No? First, you will be given easy practices. First, see, you will be given additions, deletions, subtractions, isn't it? First, you will be trained to do easy methods. And this is the microbiology which will give you the platform to understand all the biological process, genetics, biochemistry, or you name any subject of biotechnology. Actually, we are part and parcel of biotechnology. and it is a super specialized. Microbiology and genetics are the super specialization subjects of biotechnology. Biotechnology. What is biotechnology?
Combination of microbiology and genetics.
Oh, it's not biochemistry. It is not statistics. It is not zoology.
Sir,
Maximum
production of essential products through the microbes. The products which we need, we produce through
It is
microbes.
not. See, if you are using biological processes for getting some technology, is called as biotechnology. If you are using biology, it could be genetics, it could be microbiology, it could be biochemistry, it could be anything of life sciences, or botany, or zoology, or anything, cytogenetics, or environmental sciences, anything. If you are using for getting a technology or process for well-being of mankind, that is called as, for making your life easy, something is used from which is living, is called as biotechnology. In that one, microbial biotechnology, actually it plays very important role, vital role, important. So many of the processes for man, many process, see, antibiotics are being produced by microorganisms. Lactic, as there are several organic solvents are being produced. Beverages are being produced, beverages, alcohol, there is a beer, wine, and brandy, all those things are being produced. You name any product. Vitamin B12. See, it can be vitamin B12 nowadays. Vitamin B12 and D3 are very, very vital and very important issues. Lot of people are having deficiency of B12 and they are facing. See, actually, B12 is required. What is the function of B12? Can anybody mention? What is the function of vitamin B12? Hmm? It is, see, it is so simple. It is required for hemoglobin synthesis. It is required for
Cyanocobalamin, sir.
cyanocobalamin. That is a precursor. That is vitamin B12 precursor. So vitamin B12 is required for RBC synthesis. Along with it is required for regeneration of tissue. It is required for regeneration of tissue. So means cells to be formed. You name any cell in our body, you require B12. And if the see, cardiac cells does not grow. Cardiac cells does not one does will become will not become two. Even the nerve cells also, but their repair is required. Their continuous repair is required, and this is being achieved with B12. And because of this, because of lack of vitamin B12 and D3, we are getting a lot of nowadays a lot of Alzheimer's and cardiac diseases are coming because of vitamin B12 and D3 deficiency. And vitamin B12 is absolutely produced by microorganisms. No chemical synthesis mode is available. Like several antibiotics, they are produced by microorganisms as well as synthetic mode also. But this is the where only your microorganisms can synthesize. Propionobacterium shermani. There is a bacteria, Propionobacterium shermani. You know, you'll be getting. You are all young students. You must be having the pimples at your face, and there is a bacteria which is known as Propionobacterium acne. Propionobacterium acne, and it is sister concern bacteria. The genus is same, the species is different. Propionobacterium shermani produces the what? Your
Cheese.
B12. Yeah, cheese is also being produced with Propionobacterium. This and you know Streptococcus. There is a Streptomyces. There is a group of bacteria which are called as Streptomyces. These actually they are called as Actinomyces group. Classification we'll see some other time. Today's session is for interaction and subject motivation. So subject will do some other time. So these Streptomyces bacteria, they will produce this antibiotics as well as this B12. So there are so many bacteria which are producing this vitamin B12 or useful bacteria. See, E. coli is a lot of bacteria. So why you are getting a deficiency of this bacteria? Why we are getting a deficiency of this bacteria? Because the food habits and there's the antibiotic interventions that we are doing. What we do, we'll be taking the antibiotics, so that antibiotics will be removing the pathogenic bacteria. Along with that one, the good bacteria also eliminated. So the balance is lost. So due to loss of this healthy bacteria, their there's a good functions are not met. Okay, so as I told you, the microbes serves as a first tool in technology development, biotechnology, recombinant DNA technology, or any kind of thing. See, coronavirus vaccine, just coronavirus vaccine. Of course, the coronavirus is also microorganism, the virus. So, if you want to develop a vaccine, first its a gene should be cloned in a bacteria, isn't it? Because I told you, bacteria got less number of genes, so that can be easily manipulated. As you have less number of genes, that can be easily manipulated. Cut, paste, copy, that can be done very easily. Or you name like a golden rice. You must be aware about golden rice.
Yes, sir.
What is that golden rice?
Rich in vitamin A.
Good.
Rich in vitamin A.
Yeah, it is actually it is a beta carotene. It is rich in beta carotene, which is a precursor of vitamin A. Vitamin A is retinol. It is actually unstable. Retinol is a very quite unstable form. So we should not we should be taking in the form of, there is a beta carotene. Beta carotene is converted into vitamin A, and this vitamin A gene is taken out from algae. There is one of the algae, it has been taken out, and after taking it out, first it was cloned in E. coli, and that gene was then in turn kept into rice genome, along with the rice genome. So whenever the rice starts growing, its a promoter is there's a milking gene. So whenever this beta carotene is not produced as and when the seed is growing, it grows only when the fertilization is over, and when the milking or when the starch is being accumulated in the seed. At that time, beta carotene is produced and it is being accumulated over there. So here the microorganisms are involved. So first to the cloned gene, first to get the whatever the gene that you got, first you have cloned into that one. And see, suppose if you want to study a gene, whether it gets expressed or not, first that whatever the gene that you are getting from green algae, that is first kept in E. coli and that it is studied. And physiology, suppose you are keen to understand Gout's disease or any physiological Gout's, hope you understand, you know that Gout's disease.
Yes, sir.
What is that Gout disease?
Sir, there's an inflammation of the joints due to uric acid crystal, due to the accumulation in the joints.
Yeah, it is basically improper metabolism of purines and pyrimidines, so that uric acid, whatever it is produced, it is not further metabolized. Urease enzyme is missing in those people. So whatever the metabolic pathway, it is not completed in that case. So see that one particular gene, if you clone in microorganisms, if you clone that gene, and then if you put that gene into that person, then if that is taken, many such kind of thing. Gout is the first case actually. The urease enzyme has been gene has been cloned in microorganisms, first in E. coli, then in yeast, and then it was given to the patients of gout, and that whatever the missing path is there, that is completed, and the disease is there's inflammation or whatever the problems are there, that can be overcome. Okay, that was the beauty of microorganisms. And see, the DNA structure is concerned. Who worked out on DNA structure?
Watson and Crick, sir.
Watson and Crick, actually. Okay, Watson and Crick. How you will get?
Watson
See,
and Crick.
actually, experiments are concerned. They have done a model. Watson Crick prepared a model. Okay, model of DNA double helix structure. Actually, the work with on DNA was done by Emil Misher and there's Griffith. Griffith's work proved that DNA is a genetical material, and Hirsch and Chase did experiment on phages, bacteriophages, and so lot of bacterial support required. It helped this Watson and Crick to conclude that DNA is a genetical and material, and it got a double helical structure. What I mean to say is bacteria serves as a tool for various biotechnological techniques, for various processes, in fact, for various physical events also, physics events also. Biosensors are nowadays a hot topic. In order to find, see, some drones are moving, or some rockets are moving. In order to find them, we are now preparing the biosensors. See, little amount of deviation from, that is falling on this bacteria will be identified in terms of electron output. If the light is falling on it, then it will give the output in the form of electrons. If suppose drone is moving from that area, so the light which is there, which is coming on this bacteria, will be interrupted. So the electron emitting out from it will be disturbed. That's how the bacterial US Army defense people are working a lot on this bacterial biosensors to detect the drones and enemy radars and other things. So the microorganisms, even though they are very small, easy to grow, easy to access their genetical makeup, easy to access their metabolic pathways, it is very easy to understand what is happening in that one, and it is very easy to manipulate as you wish. Citric acid. Citric acid? How many of you tested citric acid? Akshita? Have you tested citric acid anytime?
It means in taste, like.
Have you tested citric acid or not? That is my question.
Yes, sir.
When?
Not in a laboratory way, sir. It's like general thing.
Where, ma?
Not in the lab or something.
No, and like in which form you have tasted the citric acid?
In the form of lemon.
Lemon. Okay, that's good. But see, all the syrups that what you are consuming, all the syrups that you are taking, the bitter taste that is coming because of the sour taste that you are getting because of citric acid. Citric acid is a good preservative. Just just recycle your knowledge about TCA cycle, Krebs cycle. Just recollect your knowledge. So there are so many acids in that one: oxaloacetic acid, ketoglutaric acid, okay, and fumaric acid. So many acids are there. They are cocoa. They are playing, isn't it? Am I right? They are playing one by one. One goes and another comes. Just their oxaloacetic acid that will enter into by taking the carbon dioxide. Acetyl coenzyme A will bind with carbon dioxide. Oxaloacetic acid is produced, and two oxaloacetic acids combines to give a citric acid, isn't it? And citric acid is subsequently further it will be converted into some other alpha ketoglutaric acid. Okay, but see here what happened. Citric acid is a so important thing. It is required for all formulation or syrups and other things. So I need to produce it in larger quantity. I want to produce it in larger quantity. So if suppose in TCA cycle, if I am disturbing, that's a TCA, there's a cycle. What happened if I am making more amount of citric acid to be accumulated, then the alpha ketoglutarate acid will be reduced, and alpha ketoglutarate acid is, and subsequently whatever the remaining cocoa players are there, all of them will become stagnant. But with genetic engineering, with microorganisms, see, we have achieved this. See, very little amount of citric acid will be converted into remaining products, and major fraction will be accumulated. This is done with the Aspergillus niger. Aspergillus, this is a black mold, which generally grows on your bread. There are so many things that grows on bread, even agaricus and rhizopus are also there. Okay, so with microbiology, with the knowledge of what happened? This girl is coming and leaving. With the knowledge of microbiology, you can manipulate the genes. You can manipulate the genes. See, from citric acid, you are getting alpha ketoglutarate acid. So if you are completely blocking this, just I want alpha citric acid, I don't want alpha ketoglutarate acid, the cell will not survive. Cell has to survive, no? So for which, what you need to do, you need to divert a part of citric acid, very little amount, into alpha ketoglutarate acid. So the remaining cycle, cocoa cycle, should be continued, and remaining 80% or whatever the remaining portion is there, that is useful for your commercial application. That can be easily done with microbiology. With knowledge of there's a gene manipulation with microorganism, that can be achieved. That is very easy to achieve with microbial skills, whereas it may not be possible with plant. If you are working with animals, that may not be possible. That is the beauty of microorganisms. And see, all molecular biology techniques, PCR or recombinant DNA technology, restriction digestions, all the techniques were developed by using your microorganisms. Gene structures were studied by using the microorganisms. So if you want to get mastery in any subject, you name botany, zoology, chemistry, genetics, sorry, not chemistry, biochemistry, genetics, any field, if you want to become a master, good head grip, you should be master at microbes first. First, you should be able to understand the microbes, and you should be able to manipulate. First, see, nobody will do the manipulation of animals or plants. First, they need to do with bacteria, microorganisms, then they should go with the higher organisms. Always we learn some basic examples, no? Then we will go for the higher things, for which you have to learn the microorganisms. If you are able to learn the microorganisms, culturing of microorganisms, then stem cells, cell lines growing will become easy, which is actually pioneering. By the time you become masters, or not even masters, by the time you complete, probably few of you complete the PhDs, even cell lines, stem cell lines may be so easy. So for which you need to have your hands good practice by using the microorganisms first. First, you should be thorough. First, you handle better your microorganisms, and which are very friendly for you. And see, they are ready for giving any product. Actually, on this earth, there are many microorganisms. And from the total microorganisms, it is believed that only around 1% microorganisms are cultured. Only 1%. And 99% microorganisms are not yet cultured. They are not yet cultured. There are so many things, they are waiting for young minds like you. Just come and explore. See, last 2 years back, tobramycin is the one antibiotic which was discovered from uncultured microorganisms. DNA was isolated from hot spring, and the DNA was cloned, and a new antibiotic, peptide antibiotic, was discovered, tobramycin, which cures tuberculosis. Like there are several things to come into the picture. And from this 1% of microorganisms, and only 1%, from that 1% of microorganisms are explored, being used for various applications. Means whatever the total microorganisms are there, from that 1.1% of the microbes are being explored for various applications: beer production, antibiotic production, acetone production, organic acid productions, paint productions, and even artificial honey preparation, and mushroom cultivation. You name any applications, microbe says that I'm here. Only 0.1% of the microbes are doing these many jobs. If young minds like you, if you start working on them, and if you explore another 0.1%, just imagine where will be the microbes. You will be having huge potential. Even the pathogenic microorganisms also. See, there are few microorganisms which creates harmful. Microbiology deals with small organisms which cannot be seen through the naked eyes. In microbiology, we will be studying about bacteria, fungi, algae, next protozoans, helminths, viruses. These are the things that what we cover in microbiology. Okay, and but more of part will be going with bacteria. Bacteria also different types and all those things. So these bacteria, harmful bacteria, they are very few in number. See, the smallpox used to be a big burden in olden days. Generally, there's kingdoms. If one fellow gets earlier, we don't used to have a very, like in very old days I'm talking about. If suppose one person gets infected with smallpox, once he dies, they will be covering the person in a blanket, and they will be taking that person and throwing him into the enemy territory, where it is a bioterrorism actually. It is now, even today, that is being practiced, but not chickenpox, smallpox kind of thing, but influenza virus in Indo-Pak border, Indo-China border. This is routinely practiced. What happens? There's influenza virus, the other person will release into the opposite territory. What happened? Influenza virus, it does not kills, but it makes you weak. So you cannot at least fight with your gun for a month or so. And even this coronavirus was believed to be one bioterrorism. We don't know how much it is true, but see, as a microbiologist, what I can say is it is not an artificially created lab virus, but it is a fusioned one. But fusions are possible in normal case also. Okay, many novel coronaviruses are many variants, many mutants are being developed. So as a microbiologist, you are having a lot of scope to work. It is a hot cake job now. The person who works on viruses, the person who works on RNA based work is a hot cake now. Scientists are preferring who works on these areas, very upcoming and very interesting areas. So you should choose that area, those areas. One like our founder of microbiology at Usman University, Polasa sir, he worked on smallpox, and in fact he got infected with this. He got a lot of blisters on his face, and it is not impossible. See, we should take it as a challenge. If you are scared, see, we are using HIV virus. After removing the pathogenic genes, its backbone, it is being used as a vector for vaccine development. There is one group of scientists who says that novel coronavirus is actually vector which was developed for curing the HIV virus. There are so many concepts. Even adenoviruses, influenza viruses, we are using them as a tool for the vaccine development. Nothing is waste here. Even pathogenic microorganism is also not a waste thing. It is also useful. But when it crosses its limit, see, microorganisms are very much friendly for us. normally when we travel in a bus, the side by like the adjacent person, the person who is sitting front or back, he must be having a tuberculosis, TB. So when he is coughing, what happens? Few bacteria will be released into the atmosphere, isn't it? And obviously, the few of them will be entering into you also. Are you getting disease? No. Generally, what happens here? There is a defense system in our body. Whenever the pathogens that they enters, their number is less, then you will not get any disease. Here also, novel coronavirus also, the virus is almost spread in the environment, in the air, but everybody are not getting disease because the level of microorganisms, that virus that they enters is important. We have a defense system which is the immune system, it is called as. So if you, again, immunology is a part of microbiology. Immunology is also a part of microbiology. So these microorganisms, our body is having a protection immune system. Whenever even the microorganisms is also having immune system. Microorganism, E. coli is having good immune system. We'll see about it later. So when the number of microbes are less, what happened? It does not create any problem to you when you work in when you start working with microorganisms. So the bacteria will enter in you, but when the small number of bacteria enters, nothing is going to happen. Most of the time we'll be working with safe microorganisms, not a pathogenic microorganisms. Unless and until medical microbiology, most of the times we'll be working with grass status, generally regarded as safe, those microbes only. But in medical microbiology, we'll be handling the pathogens also with precautions. So see, even this grass status microorganism, E. coli is a grass status microorganism. Even lactobacillus, lactobacillus is there in your curd, isn't it? So that is not a harmful bacteria normally, but in immunocompromised persons, like a patient is on medicines for a long period, or a person's kidney got transplanted, if a person is HIV positive, they are immunocompromised, means their immune system is not working properly. In that case, even that your beneficial microorganisms also will create a problem to you. See, just an example, if you are taking a cat and beating it in a closed wall room, what happened? Even it will attack after some time. The same thing is happening here. Microorganisms are not at all harmful majority of the time if they are like gone in a small quantity. But if you are taking the microbes at larger quantity, if the huge number of microorganisms are entering into your body, then what happens? Then there is a possibility of you getting the disease. So there is a rule of microbiology: whenever you handle a microorganism, you handle with safety. So whenever you are getting it sample, you treat it as a HIV positive. When you are feeling that it is HIV positive, you will be taking extra precaution, isn't it? You will be taking extra precaution. You will be doing extra precautions, and you will be careful. You will be wearing the gloves. You will be putting a mask, and you will be maintaining it as distance as possible. So you treat every sample in the same way. So the chances of getting even the one number of microbes in your body is less. You will not get any disease. So far 20 years of 20 plus 2, 22 years of my microbiology working, I have never seen any person got exposed and got diseased due to the microorganisms in the lab. That is because of the working efficiency. Please, you practice that one. So microbes are not dangerous at all if you are handling them good. But even though best organisms like E. coli is best for us, but it may also be dangerous if you are immunocompromised. You are getting my point? Harmful microorganisms are also not harmful if you are handling them properly. The friendly microorganisms also may be harmful if you didn't handle them in a properly. Okay, they gives you lot of you name anything, artificial, this American soldiers, they have prepared bulletproof jacket. They have cloned the genes of there's a fibrin gene of spider, and they have cloned in E. coli first, then into cow milk, and with the milk they have produced with the fibrin of the spider, they have produced bulletproof jacket. And the technology is microbes. Microbes are involved. Any process, microorganisms are involved. Plastic degradation, environmental cleaning, and there's a nitrogen fixation, and providing nutrients to the plants. For many applications, we require microorganisms. So, and because of viruses, as the viruses are coming in future, the virology and virus-based research is a huge topic. We're having a lot of scope for this. In fact, earlier we used to have like epidemics. Now and then epidemics used to come and go, so we never used to focus on that area, so it was not much researched. Not we don't have many virology labs. Only one Pune lab used to be there. Now I think 2-3 labs have been established. But who has to work in the lab? It is a microbiologist. It is we people have to work in that one. So you need to train. See, when you are working with a tiger, you should work very carefully. If there is any mishap, then what happened? It will attack on you. Same thing, novel coronavirus. You should work with. There is always you will be having a privilege to work with novel coronavirus, corona warriors kind of thing. In future, many viruses are yet to come. Just we are scared of one. See, bats harbors millions of viruses similar to novel coronavirus. Many things are coming. So we are having a lot of demand. And see, as a placement officer, let me tell you, we do place this microbiologists are placed in industries, in clinical side, in research side, in teaching, and the microbiologist, quality control, quality assurance, and production. These are the sites where the microbiologists are appointed, and almost 200 to 300 positions are there still for the microbiology people. We are not getting in a single person. This is to tell you the importance of the subject in industrial sector. But you should have the skills. I told you in the beginning, you should be marketable product. With the skills only you will be placed. Otherwise, without skills, I'm having a degree of 90%. Nobody will help you. I'm 90% with Dr. Pasha student. Nobody will help you. The skills should speak. The skills should speak what you are. And as I told you, many subjects were developed from the microbiology. And as a microbiologist, and last decade, it used to be the genomics and proteomics. Lot of genomics is understanding of genomes, proteomics is understanding of proteins, and now next future is about transcriptomics. There is RNA based work, viruses, understanding the viruses, and there's RNA based disease detection. All those things are there. So if you can learn more, if you can have a command with microorganisms and their techniques, if you can have good command over the microorganisms, just you'll be easy to handle the microorganisms and manipulation them. Like it is very removing of gene and putting a gene. If you are familiar with them, next, so that definitely you can conquer the next decades. This is how you can the subject microbiology is concerned. It is a very important subject. You can get a lot of lead. Just you collect one pinch of soil sample from anywhere, that you will be having millions of microorganisms, which will give you the support for doing the work. And because they are the small tools, they will support us for understanding the larger techniques which are required for the man. Okay. With this is my planned session for today. And normal microbiology introduction I will do probably in the next class. And our two teachers also will take you the classes. I think next Tuesday we are having a class 9 to 10. I'll do a introduction. There is a basic. What is the microbiology? I'll start the basic class. This is a mentoring. This 2 hours I thought of doing a mentoring. Then I'll go with the regular classes. So next class. So see, you must be having my WhatsApp number. Anytime if you have any queries, any issues, just post into it. And the syllabus immediately once the class is over, I will post the syllabus. And the books we are having the books. Even my own written books are there. This is the first semester book. Okay, you can have it once you come to the college. I'll provide you this book, and meanwhile you can have online information and try to get some presentations, YouTube channels, and go through. And we're having a YouTube channel. I'll provide you a link of YouTube channel also. Subscribe to it and see. There are some videos about the practicals of microbiology. Just see that those things, and you will be able to understand something. So just I'm stopping the recording.